Yeah.
Okay. Hi, everyone. Wow. Let's see. How many of you from my class? Good amount. Okay, you all get A's automatically. So today we have a treat. We have Dmitri Chklovskii from Flatiron Institute at NYU. So for those of you that don't know, Dmitri is a group leader at the Center for Computational Neuroscience at Flatiron, where he does awesome work on theory, computation, and data-driven approaches to reverse engineering how the brain works. Before that, he ran a group at Genelia, where he led, which at the time was the largest connectome project. So he spearheaded that and ran that at first. He was also an assistant professor at Cold Spring Harbor Lab. I actually have a special connection to Nitya because when I was transitioning from physics to neuroscience as an undergrad, I asked my professors for papers that explored like principles in brain. And someone recommended me one of Nitya's early papers on wiring optimality principles. So how the cost of axon length introduces optimality principles in neuroscience. And that was very influential for me. It gave me a first taste of what a principle of neuroscience could look like. So I know you're not here to hear Guy speak. So without further ado, let's welcome Dmitri Chklovskii.
Thanks, Leah. Can you hear me? Yes. Thanks, Leah, for a kind invitation. I have to say I'm very glad to see that you hired Leah. Because I already learned a lot from him. And one of the things I learned is that, if your name is hard to pronounce, you help people. Okay? So the field of computational neuroscience, I think, is in crisis. And of course, it's not our fault. It's the fault of the experimentalists because they've developed astonishing new techniques that produce breathtaking data sets, from large-scale recordings of neural population activity to synapse-level connectomes, and even develop previously unimaginably ways to perturb the circuits through genetic tricks or molecular perturbations or optogenetics. But all these advances create an urgent need for models that can interface with such data sets. Yet our current tools fall short. I think you will agree with me that biophysical simulations on the Hodgkin-Huxley level are way too detailed because they rely on an unimaginable number of unknown parameters that cannot be all measured at the same time. While deep learning networks, for all their success, lack biological realism and interpretability. So today, what I'm going to tell you about our attempt to bridge this gap and propose a new workable model of a neuron. Of course, if we're successful, this also suggests a new path towards biologically inspired AI. So the key to our approach is a realization that the neuron, or the brain, or the neuron, has to deal with the environment, which can be modeled, we think. If I figure out how to advance the slide. Oh, I see. Sorry. Which can be modeled as a dynamical system. Okay? Say in general, stochastic nonlinear. But of course, that is the generative system, and the state, it may not be fully observed. And so with our sensory organs, we just get some kind of observation time series, which has to then be used by the neurons to predict the future and control the environment, influence the future. And what the big question is, I think, is what is that algorithm that the neurons are performing that can solve this task. Okay? Now, I started by formulating this problem in terms of the brain as a whole, and then kind of shifted towards the neurons. And most of the talk, I will stick with individual neurons, but I will think mostly about the neurons closer to the sensory periphery. So, okay, that's where we're going to address this issue. If you have any questions, please feel free to interrupt me at any time. Okay? So here's the outline of the talk. We will try to deal with nonlinear dynamics by first reducing to linear, and then we will show how learning can be done from actual data, make some predictions, test them, use the new model to develop deep networks, and then actually make a connection with the control side of things. So nonlinear dynamical system. So I said, okay, stochastic nonlinear dynamical system. We'll simplify here a little bit and just deal with the deterministic ones. Pretty much everything I'm going to say applies to stochastic. We verified it. It's now published. But I don't want, for interest of clarity, let's just think about deterministic. So one of the standard examples is a pendulum. Of course, you know that it can be described by these equations. And if the goal of a neuron is to learn the dynamics, of course, the neuron cannot possibly learn it in a symbolic representation like this. Okay, what other representations are there? There's something called a phase portrait, which is this plot where you have the state variables x1 and x2. Of course, in the case of pendulum, it's the angle of the displacement, and x2 is the angular velocity. And in that space, you can have a vector field that, for any given initial state, will predict the future trajectory as far as you want. Okay? Now, of course, it's silly to think that a neuron could learn this whole phase portrait. Of course, there's a lot of information here. But maybe if it could just capture the distinction between the oscillatory orbit and the rotational orbit, that's something a neuron can do. And in fact, this is where most of the entropy, so to say, is produced, because if you see the pendulum coming up like here, okay, and you have to decide whether it goes over the top or comes back down, that's a big, sort of two big choices for the future, and that's where entropy is generated. So the question is, maybe a neuron can predict for a given, observing a given trajectory for some time, or seeing the state in the deterministic case, can predict whether it will continue going into the rotational orbit or turn back into the oscillatory orbit. Of course, the fate of the pendulum is determined by the fixed point. In this case, it's a saddle point, which means that this is the unstable direction, and this is the stable direction. And if the trajectory is to the left of the stable direction, so to say, it will go into the rotational orbit. And if it's to the right, it will go into the oscillatory orbit. So if only the neuron could make this call, it would already be able to predict something about the future. Of course, in general, again, this is nonlinear dynamics, but if we're only interested about the settle, what happens around the settle point, we can linearize the system. And I'm replotting here linear approximation to the dynamics around the settle point given by this equation. And then you have this vectors here, which you can think about as left eigenvectors of dynamics, or singular vectors. I'll show you in a moment. Single vectors are more convenient. And basically, you can make a prediction, given the state xt, whether it will go left or right, by just computing a dot product of xt with this vector v1, which is normal to the stable direction. Now, if this was normal dynamics, then these two lines would be orthogonal to each other. And then, of course, V1 would be just unstable direction. For non-normal dynamics, it's a somewhat more general case. And so you just keep the orthogonality property, which survives in this context. If you don't know what non-normal is, don't worry about it. Okay? So mathematically what happened here, you can think of basically describing the evolution of the state as the solution of this linear differential equation, which you know is an exponential. And this matrix is an evolution operator, so to say. It advances the state by time tau. And then what we are doing here, the intuition that I described, is simply the rank 1 decomposition of this evolution operator into this singular vectors, left and right singular vectors u1 and v1. Okay? If this is a little bit unfamiliar, think about principal component analysis. Okay? That's what this is, right? So you're just reducing all the variance of the data to a rank 1 approximation by taking the top principal component. And in this case, the sign of the projection, okay, which is, dot product of v1 and x, as we were saying, which I will call z, determines whether it goes left or right. Okay? So for the students in the audience, if the dot product of xt and v1 is positive, will it go left or right?
Those A's are dropping to B's.
Okay, let's think together, right? X dot product with v1 is positive, means that this angle is big or small? Small. That means that x is on which side? Okay, so we're going where? Great. Okay, so that's basically it. And now we need to bring it back to neurons. Okay? And the neurons, as you know, love computing dot products, right? So you take the state, you represent it in the upstream activities of the neurons, and then you compute the weighted sum, which is nothing other than the dot product between the state and this vector v1, as long as the synaptic weight vectors encode that vector v1. So it seems that the neurons can naturally compute this prediction whether the system will go left or right at the settle point by determining the sign of that weighted sum. Okay? Good. So that's all that we need to know about the linear dynamical system. But of course, the environment is a nonlinear dynamical system. So what happens there? Well, let me just redraw the results we just got. It's again the zone around the settle point where the dynamics are linear. And what I plotted here in color is the value of that dot product, positive to this side, negative to this side, right? And if you came from this yellow region, you will turn left, as we just figured out. If you came in from the purple region, you'll turn right. Okay? So that's all you need to know from this linear analysis. But there is something else in the linear analysis, which is you can see that there is a symmetry that you can just flip the sign and go between the two sides, which is, of course, has to do with the fact that the x dot equals grad f x is a linear system and it possesses this symmetry, x to minus x. Okay? But the general nonlinear dynamical system doesn't have that symmetry because outside of this linear zone, this so-called invariant subspaces become invariant manifolds, lead you to another settle points perhaps, and there is no symmetry between this settle point and that settle point. They're completely distinct. Okay, pendulum is a notable exception because there's just one settle point. Okay, you couldn't have them distinct. But in general, there is no relation between these two futures. So having the symmetry is actually a nuisance. And to express it in the neuroscience terms, you know, when you're predicting the future, say to make a fight or flight decision, which is how you can think about this point, the two behaviors that go with this decision are not mirror images of each other, right? Fight and flight invoke different muscle groups. And so you can't just flip the sign and switch fight to flight, right? That's the nonlinearity of the whole thing. And so instead of just switching the sign, what you need to route the nerve signal, the signals along the nerves to different recipients, to different muscle groups, right? Like arm versus legs and so on. Okay? So then instead of having one neuron that outputs positive and negative versions of dot product, we would want to have two neurons, each of which computes the same dot product, but then rectifies them in a different direction. So this neuron will output a non-zero signal when the dot product is positive and the system is going to the left. And it will output 0 in the opposite case, while this neuron will output a non-zero signal when the dot product is negative, and then 0 in the opposite case. Okay? And then these two neurons can project. They receive the same input x, but then they can project to different muscle groups. So that's basically why we think there should be rectification and not just the weighted summation. So to summarize it, we're thinking of neurons as being detectors of the dynamical trajectories around settle points, where the prediction is made by the projection or dot product of the state onto this vector v1, followed by the sign function or rectification. Any questions?
Is V fixed?
Right. So that's a very good question. So V here is viewed as a right singular vector of the linearized dynamics matrix of this evolution operator. So if I tell you the dynamics, you can find out the V and encode it as synaptic weights. But of course, there is no possible way that a neuron knows the dynamics. And so for this model to be workable or plausible model of biological neurons, V has to be learned. And then it just tracks the natural stimulus statistics. Is that what you were asking? Any other questions? What will the neurons do?
Yeah, what will they be communicating? Will they be throwing
Nothing.
the system?
I think that's what the receptive field is, right?
I see. So you're counting on they're only gonna get input when they're near the
They will get input at all times, but it will not be aligned with their synaptic weight vector.
And what if the system is just far from the settle? What is they still have a vector. They can still do that dot product.
Right. So this is an excellent question, and I have to admit that we don't completely understand it, because in traditional dynamics, you would have two vectors. The location, the state corresponding to the settle point, and this Lyapunov, say, vector in the tangent space. Now, the neurons don't have the luxury of representing two vectors, right? There's only one synaptic weight. So somehow there has to be just one vector that they use. And perhaps with rectification, this could be understood as some property of a very high dimensional space where most of the interest in dynamics happens on the thin shell at a fixed radius. And then you just have one vector that determines the receptive field and the kind of the future prediction. Does that help? I mean, I have to admit that this is something we're trying to work out, but that's my intuition. Okay.
Are you trying to make a claim about the nature of all neurons?
Yes.
Or there are some neurons that are particularly looking out and coding?
So we will apply this primarily to the sensory neurons.
Okay.
But the main reason for this, I think, is because we can control the stimulus. And otherwise, you don't know what they're getting deep in the brain. And the lack of feedback loops. Because what I'm going to tell you isn't yet advanced enough to deal with feedback loops, which is OK where you're at the sensory periphery. But it gets troublesome when you get deeper into the region.
Right, but I guess the broader question I'm asking is, can you just do with populations of neurons that are tuned to the saddle points in the dynamics of the environment?
Right,
Ever
but what's the environment? So I would claim that the neurons deeper in the brain are tuned to the settle points in the dynamics of their environment, which includes other neurons, right? So each neuron is kind of looking out for its favorite settle point.
Is there some sense in which, like, there has to be that there's a space of saddle points that has to be covered or tiled?
Yeah, I mean, you just have to have, at least 2 neurons per settle point, and then, but there are a lot of neurons, so I think it's not that bad.
Okay.
Any other questions? Okay, so we have this model, right? But as Leo insightfully pointed out, it's only valuable if we know the value of V, and it stays constant for some time, right? Where does V come from? And so what we're going to do, we're going to fall back on actually existing method that allows you to learn V directly from the data. So that's the setting that is more appropriate for neurons, because nobody told the neuron what that function f or gradient f is, right? And instead, it just sees the trajectories, one after another, which are communicated by the sensory organs. Can you learn that vector v from trajectories? And it turns out that this can be done, and I'll show it on the example of a scalar time series. So to simplify a problem, now we will just think about a neuron that has only one synapse, okay? And it gets in a scalar time series. Such neurons actually exist. These are neurons that are in our eyes and in invertebrate eyes, as I will appeal to, right behind photoreceptors. So think of this as a photoluminance signal that comes from photoreceptors. And the photoreceptors produce this series because they basically just scan the visual scene due to self-motion. And so we took natural scene photographs, scanned them like this, produced images like this. This is actually a synthetic one, but the results don't depend very much. Like any kind of like natural scene will give you this results, okay? But if it's a scalar time series, you would say, what kind of settle point is there, right? It's a one-dimensional dynamics. Well, but my contention is that it's only a partially observed system. And so the dynamics is higher dimensional than 1, but you consider only a scalar projection. Okay, but then how do you get the state vector? Well, so that's well established in dynamical systems. You do time delay embedding. What that means is that you do the measurement not just at one time point, but over a window of time, like shown here in blue for the past or red for the future. And that gives you a workable representation of the state. It may not be a parsimonious one, but it is a full dynamical state in the sense that it can predict the future of the system if the lag vector is long enough. And you can appeal to things like Tekin's theorem to justify that. So then what we are doing here is we want to make a prediction about xt plus tau, which is the evolved version of xt. And you can, of course, do that by computing the canonical correlation analysis between those two data sets. Okay, so where do those data sets come from? I have the past and the future window. I slide them across the data set, across the time series, and then I get two corresponding data sets on which I can do canonical correlation analysis. Then the canonical direction, which is what, which is closely related to that vector v, gives you that vector v1 that I was telling you about, and about which needs to be encoded in the synaptic weights. Now, there's only one synapse here, so what's the synaptic weights? Well, the neurons also have temporal filters. So then if your state is represented by a lag vector, then the corresponding vector v1 will be the temporal filter that is convolved with this time series. So when we do that, when we do that, we get shapes like this. So the way this is organized, this is kind of the present, like this point here. And this is the filter that we learned from those covariances in this data set that actually corresponds to the second singular value. Just it's better for comparing to the particular experimental data I'll show. And that is kind of like a smooth derivative filter. And it, of course, looks like what you get from post-photoreceptor neurons like bipolar cells in vertebrates, or so-called L1 and L2 cells in flies. So you reproduce this biphasic structure. What you don't get is that this singular vector v2 does not drop down to 0. But I think that's just the consequence of the fact that this filter, the temporal filter in neurons, has to be computed by a physical system that is causal and continuous. And so the filter cannot reach forward beyond 0 because then it would be non-causal. So that means that it had to be, start from 0 here and be continuous, and that's what gives it shape. These constraints are not put in here for simplicity. So that's why you kind of see this disconnect. But other than that, we seem to match the shape of this blue line with the black line. Any questions about this?
How is this CC analysis relates back to the fixed point decomposition at that point?
So this vector v corresponds to the vector that is orthogonal to that stable direction.
Without even knowing the environmental dynamics F?
Without knowing the environmental dynamics F, because it's implicitly encoded in this evolution, right? So to get from this state vector to that vector, you need to apply that evolution operator, e to the gradient f tau, okay? So you can, instead of knowing f, you can do the single vector decomposition of that operator by looking at the covariances of those. Does it make sense? Okay. Any other questions? Please.
So if you have multiple such dynamics, is there a way to learn how to not interfere with the V that was learned from one neuron to then recruit other neurons?
Yeah, so I think that's somewhat related to Karina's question. That neurons, we know from neuroscience they have receptive fields. So they kind of have local responsibilities. You can get that from rectification, of course, depending on the bias. So that they kind of probably tile the space. Then this learning is completely self-supervised, and you just apply it to the input. But if I also introduced a factor that depends on the output of the neuron, that would confine this data on which CCA is done to the data that falls in the neighborhood of that fixed point. And we know both from Hebb and from STDP that, in fact, yes, postsynaptic activity impacts synaptic learning. So that would be my answer to that. Yeah? But we haven't put it in yet, actually. In this, what I'm showing you is just the CCA. But actually, for this bipolar cells, they don't seem to really, because they're common on and off pairs, which are these two components that I was talking about, the two rectifiers in opposite directions. In the fly, their temporal filters are very similar. So we think that on that level, it's primarily done just by CCA without using postsynaptic activity. But once you get deeper in the brain, we know for a fact that has to enter. And I'm not showing you how to do that yet. Okay? Okay, so this, this agreement with oh, by the way, I should also say that this is a future filter, which is the left singular vector. And it can be used to predict the future, like we had that vector u that showed in which direction the system will go. It will become important in a minute, but at this point, I'm just telling you that it's a pair, okay? So it's there. Now, it's nice to have this match, but it's no more than a sanity check, because there is at least a dozen predictive coding or efficient coding theories that will give you biphasic filters like this, okay? And we can also reproduce the signal-to-noise adaptation and so on, but other theories can do that too. So can there be a prediction that is specific to our theory that is really non-trivial? And we think that there is one, and it has been missed by most predictive coding theories. And that has to do with what I call predictive and retrospective neurons. And to see where this comes from, let's go back to our favorite saddle, okay? And the question we asked, if I tell you the state at time t, can you predict whether it will go right or left or right? Based on the projection, we can do that. And you project it to V1 and rectify. Okay? But I could have asked a different question. Based on the state now, can I predict where the system came from? Whether it came from this direction or from that direction. Okay? So instead of asking to predict the future from the current state, I ask you to retrospect about the past from the current state. That can be solved very similar way. All you have to do is to compute a dot product, not with V1, but with this vector U2, which is that left singular vector that I said we will use in a minute. Okay? And the sign of the projection will tell you whether you came from this yellow region from above or from the purple region from below. So I can answer this question just as I answered the prediction question, except by projecting onto a different vector. Okay? This raises two questions. One, why would anyone do that? Like, the past already happened. Okay? Let's stop obsessing about the past. Let's move forward into the future, right? Like, good advice. But imagine that your system is only partially observed and observation is noisy. Then you can improve the quality of your state inference by retrospecting. In engineering signal processing, this is known as smoothing, where you use data after the time point you care about to improve the estimate at that time point. We do it all the time. Imagine talking to someone in a noisy room. You can't quite make out what they're saying until they finish the sentence. And once the sentence is finished, you can retrospect and figure out what they said. What we are saying, also another example is, of course, saccades and blinks, where you have to fill in the gaps. And what we are saying is that that can be done on the level of individual neurons. But it cannot be the same neuron as the predictive one, because you have to project onto a different vector. And the vector are synaptic weights or temporal filters, and therefore you need two separate categories of neurons, predictive and retrospective. That's the prediction that we can test. or maybe the retrospection because the data already existed. Okay, we took this data set from this paper where they recorded from the two kinds of relay neurons, in this case, for olfactory system. They receive inputs from the olfactory receptor neurons in the mammalian olfactory bulb. They're called mitral and tufted. And they recorded their responses to white noise stimulus blindly, so they don't know which ones are mitral, which ones are tufted. But they gave us all the filters, and we analyzed them from the perspective of our theory, okay, which predicts that some filters will be orthogonal to stable subspace and some to the unstable subspace. When we talk about lag vectors in time, stable and unstable simply means exponential functions, right? Stable is a decaying exponential, unstable is a growing exponential. So what I'm telling you is that there should be neurons whose filters are orthogonal to the decaying exponential, and there should be neurons that are orthogonal to stable exponential and to unstable. And these are different kinds of neurons that have two different shapes of filters. Perhaps this one looks more familiar because the lobe preceding the spike is bigger than the lobe further back in time. That's what textbooks usually show for the retina at least. And we associate it with the predictive neurons because it's orthogonal to this stable subspace. But these also are encountered in the data set that we worked on. Maybe 1/3 of cells are like this. And here, the layer phase is actually smaller. And what we claim that these are the retrospective neurons. Where did this come from? So if we go back to this pair of filters, right, the predictive neuron would project onto the right singular vector, and the retrospective would project onto the left singular vector. And you can see that there is a bigger weight here to the left towards the present. Of course, to make the procedure physically possible, the neuron can only do that when this passed. So it's not real future. It's just a more recent past, allowing you to infer a more distant past. Okay? But that's what we are saying those neurons are doing. Another way to think of those neurons is because the white noise stimulus is not always good at eliciting neuronal response. What if you showed a step stimulus, which is often done in various sensory modalities? Then we computed this response assuming this linear filter. And these are like the standard overshoot responses. This one is an off, but doesn't matter. But this guy is weird, right? It responds with a delay and without an overshoot, and instead there's an overshoot later. So that's kind of like a crazy neuron. But that's how mitral cells really respond to stimulus. Now, if you don't care about olfaction, in case some visual chauvinist walks in, this kind of cell classification has been known, okay? So it's a retrospection, has been known in the visual system forever, okay? In multiple species. This is a relay neuron from the LGN, which receives inputs directly from retinal ganglion cells. And this is the filter of a standard predictive cell. That's the textbook example that was on the left, okay? And this is its step stimulus response. But there is also what has been called lagged cells. Of course, that's the lag to the step response. And this is not a filter. This is impulse response. So the time axis has been flipped. So here, the bigger lobe is to the right of the smaller lobe. And this is what we would call a retrospective neuron. So we think that, and they're kind of like 1/3, 2/3 in the LGN of these creatures. By the way, there's like very colorful history where it was first discovered in cats, and the serious monkey neuroscientists didn't believe it, and there was a big fight. And it turned out that actually they do exist. Any questions? Okay, so let's put together what all that I told you about, and with the eye onto the artificial neural networks now, or modeling biological networks. So this is kind of like the standard model of a neuron, which goes back to McCullough, Pitts, and Rosenblatt, and others, which is known as rectified linear units, right? Weighted summation, and then rectification, which is kind of close to what we have, right? But there are important distinctions, and that's what I want to emphasize in this slide. First of all, this is static processing, okay? That means that the neural net takes the inputs that came in at the same time, synchronously computes the output at the same time point, and then resets without memorizing the past. You may say that's not a big deal. You can do a convolution over time here. Perhaps you can. But what I think is the biggest deal is that where those weights come from. And then traditional neural nets, especially in deep neural nets, you use error backpropagation, which is a supervised method that requires ground truth labeled data and non-physical way, well, the biologically implausible way of propagating the errors. And what we propose to replace it with is what we call a rectified spectral unit, right? That's our guy that computes the dot product between the state, which has both the time index, like Lowe's-Lag vectors, can also have a spatial index I, in case there are multiple synapses. And then it's convolved with a spatiotemporal filter W, right? Which can be computed in a self-supervised fashion directly from the input data by just doing past-future canonical correlation analysis. So that's the huge difference between the two, because now we don't need backprop. We can just learn these weights locally, propagate the signal, then learn on the outputs of this layer and the next layer using the same algorithm, and so on. Does that actually work? So to test this in practice, we applied it to the fly visual system, where we happen to have the connectome, thanks to the wonderful people who worked with me at Janelia. And we specified the general architecture of the network based on what we know about the fly. And specifically, what happens in fly, they don't have just one lens eye. They have a faceted eye with Drosophila has 800 those lenses, and you can think of them as 800 pixels. And so basically, information signals from each pixel are processed independently of each other in the first two layers. So this is where those neurons, remember I mentioned L1, L2, the on and the off cells, who were biphasic, and we could learn them from natural scenes. So they sit right here. But there are other cells too, and their filters correspond to other singular vectors with opposite rectification and so on. And so we can use this general architecture and learn all the temporal filters and synaptic weights of this network without putting them in by hand or without setting any kind of supervised target, okay? Just from the translating natural images that we just take, natural image data set and scan it and present to this network, okay? So what does it do in the end? Well, in the fly, in the third layer, there are these cells so-called T4s that are directionally selective. So these are motion detectors. And these two are T4A that detects motion to the up and T4B motion down. And we saw how in the trained system, those directional selectivities emerge just in a self-supervised setting, including, as I said, both temporal filters and synaptic weights. Okay?
I think I missed how the weights W are actually updated. So
Okay.
yeah,
Yeah. So basically what we do, we take, we scan a natural image. We get a time series. We take that time series. We slide the past and the future windows along the time series. And then we form the past and the future data sets. Then we perform a CCA on those data sets. And then the canonical direction that comes out of the CCA, which is analog of the, of the loading of PCA, determines the synaptic weight vector. Okay? Now, for this, because it's a scalar input, it's just a temporal filter. For the cells that combine multiple pixels, it's a spatiotemporal filter.
And then when you actually run inference on this model, you're doing that kind of convolution with the weights that you found through the CCA thing.
Right. So what I do is the first filters of the first layer are found from the natural images, just scanning that time series. Then I compute the output by passing the signal through the dot products and rectification. And then I get the activity time series for the next layer. I apply CCA again. I learn the filters. Then I pass through those filters and rectify and get to the T4 layer. And the T4 layer does the same thing again.
How do you sample the natural images? Are they just interleaved or repeated in the same order over and over again?
I don't know if it matters. We just took a lot of photographs and just scanned row by row. We thought maybe it's important to make sure there are no jumps when you go like from one edge to the other edge, so we got rid of those. I'm not sure it matters too much. Like,
I mean, this relates to my previous question. So the more specific question is if there is some overlap, so you're still using the same receptor fields, and the dynamics are just a little bit different.
Yeah, of course. The assumption here, because we're just doing CCA on the whole data set, the assumption is that this is a representative sampling from some underlying generative process which has fixed second-order statistics, right?
So you wait for the whole data set to be done before doing this?
Well, because we're doing CCA, we don't have like an online learning rule.
You could do it on chunks of the data.
We could. And so basically we studied adaptation by looking at different things. I think the most interesting results that can be compared to experiment are signal-to-noise variations. So if you add noisy data, you get less differentiating and more integrating filters, which is like what they see. I'm not very strong prediction, but that's kind of where we got. Like you do get slightly different filters from different data sets, and we don't know how to get the real natural image data set that really represents the luminance on the photoreceptor. Like we tried several things, and they do give slightly different results. But because we don't know which one is the correct one, it's hard to compare with experiment.
I don't want to relate too much, but I'm just thinking of like in a lot of other scenarios, I'm not an expert on sensory processing, but you don't necessarily like want one model of everything at once. You want to have sort of experts that are in different kinds of dynamics, right?
So somehow they self-select, they self-differentiate in that respect. Like for example, I'm only showing you the on motion pathway. There is an off motion pathway that is originated by the cells that are called L2 that I mentioned, but they're not shown in the diagram, that like processes in its own way. So they kind of like, there is a lot of division of labor there. And that's like this rectification. You go left or right, and you do that separately. So that's as far as we can get to. Like this very interesting question of tiling that you and Karina asked about, I don't have a full answer to that. But I think that we will get it once we include the output activity in the learning rule, which is not yet included here. Okay?
I have a different question. So within like layer, do you have any interaction between neurons?
At this point, no, because CCA does that for us. They're orthogonalized. Okay. In reality, of course, it's not CCA. It's a local learning rule. So there needs to be a competition mechanism. And we developed local learning rules for that in the context of our previous work. So we know how it works. You just put one inhibitor interneuron that kind of couples them, or you have anti-Hebbian mechanism between them. So it's doable. We haven't done it yet for this problem. In the connectome, well, there is feedback. There is some feedback, absolutely. And there are lateral connections and both some feedback connections. But I think this early sensory processing stage, it's primarily feed-forward. So, yeah, so I think we're not too far off here. We are trying to build this for the case of feedback loops, but that will become, of course, more important as you march deeper into the brain. Yeah, that's a valid concern.
Just a clarifying question. Do you learn the weights in the first layer first and then?
Yes, we do. Yes, we do. I'm not sure. I'm not sure that matters, but we do this in stages. Yeah.
So what are your thoughts on how would that change if the network is like random connections? It starts from random.
Yeah, I think it will. Yeah, I think it will probably work the same. It will take longer, but in development, I mean, you can correct me if I'm wrong, but I think that there is a progression of maturation from the periphery inside. So this is not like inconsistent with biology to learn layer by layer. Okay. All right. So I don't know. I mean, are there Drosophila vision aficionados in the audience? I should probably skip next slide. It goes through all the things that our network produces, like synaptic weights compared to the ones we obtained from connectomics. They're not the same exactly, but they're in the ballpark. Sorry. This is the directional response. In the interest of time, I want to skip this. Ask me after the talk if you want to know exactly. But we even reproduce the same motion detection mechanism as has been teased out by physiology, by Michael Reiser and Axel Bor's group. And we even reproduce an optical illusion that has been shown to exist by the Clark group. So the last thing that I want to say before my time completely runs out is that I've been saying feed forward, but there are loops. Oh my God, what are we going to do with the loops? So we've been working hard in that direction. We are not yet done, but we think that what happens is that once you have a loop, it makes think of a neuron as not just input-output device, but actually a feedback controller. Okay, so instead of this picture, we have this picture in mind. Now, if this is a motor neuron, then this would be external environment and the proprioceptive feedback from the muscle. If this is a neuron somewhere deeper in the brain, then these are just like other neurons in the surrounding area of that neuron. It's neighbors, synaptic neighbors. Okay, so once you take this point of view, then neuron kind of acquires an agency. It doesn't just like transmit information, but it actually strives to steer the state of the world into some particular direction. And then, yes, it still has an input-output function, but that input-output function serves a purpose of producing certain state that is observed by the inputs of the neuron. And so the outputs of neurons then try to steer the environment to a certain state. Okay? So that's the kind of the conceptual change that we proposed. People have said, of course, that the brain as a whole does it, and the certain nuclei like oculomotor does that. But what we're saying is that it could be applicable on the level of individual neurons, right? And then it turns out that you can apply the same mathematical picture that we had with the saddle point in the context of prediction, where we had to predict left or right, but now change the task for the neuron, not just to predict whether you'll go left or right, but to make sure that you will end up on the right and not on the left. So then we pose a control problem for a neuron where the dynamics is described by this familiar linearization around the saddle point, plus the control term, which now has a particular orientation, and plus noise. Remember how I said we can do all this for stochastic dynamical system? Everything I said applies to the stochastic just as well. But this is the first time I'm just putting it in. And it still works here for control. So what do we do for control? We want to make sure that the system ends up on the right and not on the left by applying the output of the neuron, the Z, to this dynamical system. Okay? And of course, we have to minimize Z squared because otherwise, control theory is boring. You just take the state and place it there. Okay? And so this problem amazingly has an exact analytical solution. And that solution, in the limit of zero noise, is just a rectification function, which is our residue rectification. It's very easy to see intuitively why this is the case, because the only direction that matters here is along V1, right? In this direction, it doesn't really matter. But along V1, what you see is a linear force. And here the force pushes you to the left, and here the force pushes you to the right. Okay, that's the nature of this term. And so if you want to make sure you end up on the right, and you're already on the right, and there is no noise, you don't need to do anything. You just sit and wait, and the deterministic dynamics takes you there. That's this zero part. But if you're on the left, you're in trouble. If you do nothing, you will end up in the undesired state. So what do you need to do? Well, you need to compensate this repulsive force. Well, it turns out if you just compensate it, this integral gets really big. And the optimal value of this integral is achieved when the force sign is reversed. So your control is double the force on this side and nothing on this side. So we call it double or nothing control law. And this is exactly what ReLU or ResU will predict you should have. Okay? Of course, when there is stochasticity, there's finite noise, then the curve gets smoothened, and the more noise you have, the smoother it becomes. But that's basically, there is an exact solution, which I guess I forgot to paste in here, but doesn't matter. It's a bunch of Gaussian, the sort of cumulative error function integrals that you can use to compute that, which in the limit of zero noise reduces to just a rectification. So we think that this point of view of neurons as being detectors of and predictors of the future around settled points can also apply to on the control side, on the behavioral side, where neurons can perform control by producing this needle perturbations where they are the most effective. That is near the settled points where a little perturbation can influence the history a lot, and that of course ties into the point of these settled points being the point of entropy production, because where you have highest entropy production, that's where the highest of the impact control is, and this all ties in the stochastical thermodynamics picture, where optimal control relates to maximum entropy. Don't have time to go into that, but it's all different, two sides of the same coin. Okay? All right, I should conclude. So what I was hoping to communicate today is that we now have a dynamical model of a neuron, which is self-supervised, learns from the input data alone, yet is powerful enough to learn non-trivial features like motion detection, and so can be used both to model neuroscience systems and as a potential path towards next generation biologically inspired AI. And finally, the most important slide, I want to thank all the people who were involved in different aspects of this work, and I'm very privileged to work with this incredibly talented individuals. Thank you very much.
So maybe time for one or two questions. Does anybody have any? Yes.
Are you making any assumptions on how this data will work with are there any kind of dynamics that this learning can
I mean, if you run it, it always works because it's just CCA. So in that sense, it works.
So it's not based on like Markovian.
Oh, absolutely, it absolutely is. Yes, of course.
So is there any example that like this learning can learn a dynamic in the natural world?
I cannot think of any, but we don't have a theorem that says that it does either. So I don't know. I mean, there are a lot of assumptions here, of course, and we have to demonstrate that it works. I think that it's on us to show that it works in various dynamical systems, which we haven't. Yes, good point.
I was wondering, and this was fascinating.
Thank you.
The alternative principles would be things like in computer vision, like kind of next frame prediction, so straight Euclidean reconstruction of the input. I'm also thinking about things like all the work on like the 90s on slow feature analysis, or anyway. Do you have a? I mean, it's a tough question to answer, but do you have any kind of specific examples of the kinds of selectivity that you would be getting with your approach that you're going to be getting?
Yeah, so absolutely excellent question. I didn't have time to go into the related work. We have it all in the paper. But of course, slow feature analysis is closely related because basically this kind of top singular value we're looking for the ones that are closest to 1, meaning this is longest lived segregation, meaning that is the slow feature. So absolutely related to slow feature analysis. But I think the slow feature analysis kind of fell short a little bit because, although a fantastic, great contribution, it was very inspirational to me, but they didn't do the full dynamical systems analysis. They just wrote down this heuristic equation of minimizing the square of the time derivative, which is great. But the dynamical systems theory gives us more. For example, the retrospective neurons prediction wasn't there. And that needs a two-sided view. The other thing you mentioned is minimizing prediction error, which is a completely valid approach, which is very closely related, again, to the CCA, but it's not exactly the same because CCA just finds the maximum correlation direction, right? So the magnitude is not controlled. So it's not exactly the same, and we're still working through whether we can find examples where there will be distinctive predictions between the two. Thank you.
One final, very brief question, if anyone has it. Okay, if not, then let's thank our speaker.